This module is on the semi-permeable membrane. We're going to look briefly at the movement of water, and then at the movement of solutes, or the diffusion of solutes, and ions across membranes. And in fact, ions are just a special case of solutes. This should be a review from a previous course. The diffusion of water across a membrane is of course called osmosis. We'll look at animal cells to start with. Here in these beakers are some giant animal cells shown in sort of fuzzy black. On the left, the cell is swelling and then bursting. In the middle, the cell is holding its own, as we would say. And on the right, the cell is shrinking. The question is, relative to the cell, what are these solutions? Well, the solution on the left is called hypotonic to the cell, which means it has fewer or less dissolved solutes. The result is the water is going to move from the region where it's at high concentration, where there are fewer solutes, i.e. outside the cell, into the cell. It rushes into the cell, which has a higher concentration of dissolved solutes or substances. The cell, of course, swells and bursts. A cell in isotonic solution is in a solution whose solute concentration is comparable to that in the cytoplasm, and so there will be no net movement of water into or out of the cell, and so the cell will remain in its normal shape. This is a hypertonic solution in the beaker, which means the solution has a much higher salt concentration than the cytoplasm of the cell, and so the reverse of what happens in a hypotonic solution will take place. Water will attempt to leave the cell in a hopeless attempt to dilute the solute concentration in the beaker or outside the cell, and so water is lost and the cell shrinks, shrivels, and of course dies. In fact, the cells burst and die in hypotonic solutions, and animal cells shrivel and die in hypertonic solutions. Here are some coping mechanisms that cells have to deal with osmotic swelling of the sort we saw before on the left side of the last slide. Animal cells can expel ions through membrane proteins that facilitate their discharge. Plant cells, of course, have a cell wall which prevents the cell from swelling beyond a certain point. And many protozoa, that is, single-celled organisms that live in lakes and streams, actually have little contractile vacuoles that will discharge water that would otherwise continue to accumulate and cause the cell to burst. This would be, in class, grist for a clicker question. The implication of all of these observations is what exactly? That cells usually live in hypotonic environments? Do they usually live in hypertonic environments? Living in an environment, by the way, includes, if it's a cell inside your body, what is the blood or the body fluids relative to the cytoplasm of the cells that these fluids surround? Could the cells be living in an isotonic environment most of the time, but occasionally experience hypotonic environments? Is it that cells live in isotonic environments most of the time, but occasionally experience hypertonic environments? Well, the answer here is that cells generally live and must live in an isotonic environment so that they are not threatened with swelling or shrinking. The implication that cells have mechanisms to cope with osmotic swelling implies, however, that from time to time, or in some cells much of the time, cells experience hypotonic environments, that is to say, environments in which dissolved solutes are at lower concentration than they are inside the cell. Okay, now we want to focus on the movement of dissolved substances, that is, solutes, across membranes. There are several ways in which this happens. Passive diffusion, shown here, is simply the movement of a molecule directly across the phospholipid bilayer. This kind of passive diffusion is limited to small nonpolar molecules like the gases nitrogen, oxygen, carbon dioxide, some hydrophobic molecules such as steroids, which do not have trouble negotiating a hydrophobic fatty acid environment, as well as small, uncharged but perhaps somewhat polar molecules like alcohol, that is ethanol, and glycerol. Facilitated diffusion, shown here, can be of several types. This one was through a channel protein, and this one is through a carrier protein. The difference basically is that carrier proteins will generally undergo significant conformational change during transport, and that was illustrated by my wiggling receptor here. Molecules like sugars, amino acids, ions, for example, the carbonic acid ion, which leaves red blood cells and then eventually results in our expelling CO2. These are molecules that will move across membranes by facilitated diffusion. Until recently, water was thought to enter and leave cells exclusively by passive transport, but there is actually a facilitated diffusion protein called an aquaporin I forget when it was discovered, but in 2003, the Nobel Prize was awarded for the discovery of aquaporin, which is actually required to get movement of water in large amounts across membranes. Now, the important thing about diffusion, whether it's passive or facilitated, is that it occurs down a concentration gradient, or we say along a concentration gradient, by which we mean simply from a region of high to a region of low concentration. Facilitated diffusion through a carrier protein is generally slower than through a channel protein, and this is shown in the illustration here from your textbook. We can look at the kinetics of passive and facilitated diffusion and show the difference. We measure the rate of movement of the solute, the rate of crossing a membrane, say accumulation in a cell or on one side of the membrane, and the concentration of the solute that achieves that rate. So here we have simple or passive diffusion. That's the kinetics of simple diffusion. And here it is, the kinetics of facilitated diffusion. If this reminds you of something that we saw in enzyme kinetics, well then it should, because what's going on here is that facilitated diffusion is a saturable phenomenon. Why is it saturable? That is, why does the rate of diffusion reach a maximum velocity when you add high concentrations of solute to one side of a membrane? And the answer, of course, is that after you add above a certain point or concentration of solute, after you achieve that point, you can't get diffusion to occur any faster because the carrier proteins or the channel proteins are saturated with the solute. So you can recognize facilitated diffusion as a saturable phenomenon, whereas simple diffusion is not saturable. There are three modes of facilitated diffusion shown here. Uniport, the example here is of glucose passing through a glucose transporter into or out of cells. Most cells have this. Here is symport, and I'll show you this in more detail in just a moment, where glucose in kidney cells, for example, is co-transported with sodium ions. And the reason for that will become clear in a second, as I said. And then antiport, which we've actually seen in the crystal membrane of the mitochondrion. We exchange ADP with 3 negative charges for ATP with 4 negative charges. So the ATP leaves the matrix of the mitochondrion while ADP enters it. And you can see, if you remember perhaps, that this happens in part because, or it's driven in part because there is a potential difference across the membrane. There is a positive charge on the outside of the crystal membrane and a negative charge on the matrix side of the crystal membrane. And the result is that ATP with 4 negative charges would probably, if you think anthropomorphically, would prefer to go towards where there are more positive charges, and it would drive ADP accumulation by the matrix, which of course is where ADP will be phosphorylated to make ATP. Well, I'll have more to say about that in this slide. So solutes, again, diffuse through carrier or channel proteins, always down a concentration gradient. Symport and antiport that we've just seen can result in faster diffusion, and I'll show you that now. There are two modes of glucose transport, through the glucose transporter, which I showed you a moment ago, or sodium ion enhanced glucose transport. So let's take a look at these. Here we have the glucose molecules moving through a glucose transporter. It has changed shape in order to capture the glucose on one side and release it on the other side. Here is glucose transport enhanced by an electrochemical gradient. Notice again that in this cellular membrane, in this plasma membrane, the outside of the cell is positive and the inside is electronegative, so there is a membrane potential across this membrane, and that's going to allow symport of sodium ions and glucose. So this protein has evolved to take advantage of the electrochemical gradient, where the sodium ions are able to move in readily because they are positive and the inside of the cell is electronegative relative to the outside of the cell. And as the sodium ions move through this facilitated diffusion protein, they took with them the glucose molecules, and you saw it happen at a much faster rate than regular facilitated diffusion of glucose through those orange glucose transporter proteins. So there it is. Cytoplasm is negative relative to the outside of the cell, and the sodium ions are higher in concentration outside the cell than inside, which is a great setup for this electrochemical gradient to force facilitated diffusion of glucose into cells. Let's take a quick look at a voltage-gated potassium channel. This is just the structure of the protein. This is a single polypeptide with multiple helices crossing the membrane. They are stretched out here rather than folded, but when folded in the membrane, they create a pore. The domain of the pore is shown here in purple. One of the helices helix S4 doesn't show it quite here, but is a component of the pore. And if you look at the amino acid side chains here, they are highly charged, and it is as if, theoretically anyway, this helix is acting as a charged voltage sensor. In other words, it can sense differences in electrical potential which will allow ions to flow through this pore under the right conditions. And that brings us to the end of this presentation.